Hello students and viewers. Today we are going to learn second point of uh, your chapter number second, seed certification. So in a previous chapter, in a previous lecture we have learned about the concept of seed certification and procedure of seed certification. Now this is the second point of chapter number second, that is seed certification. This is point number second, field inspection. So here again we are going to deal this subchapter in detail. Now, so here what are the conditions for field inspection? How field inspection is done? What are the conditions and in which stage you have to made these field inspections? So in detail we are going to learn this field inspection point. Now see condition for field inspection. So first condition is very important. The crop should be this field inspection should be done for standing crops. So yes, compulsory standing crop should be there. The crop should not be fallen down. The crop should be standing, so that the field inspector should come and he should inspect your crops. Second, investigation and test about that particular crop seeds should be done in a laboratory. So obviously some tests are there and that should be done in a laboratory conditions. Next, objectives of field inspection. What should be the objective of this field inspection? Some objective should be there. With the without that objectives you can't make this field inspection. So see here few objectives I have placed. First objective, the seed should be grown in a field that satisfies the prescribed land requirement. Let me take this marker. So land should have, or that should satisfy the prescribed land requirement. Land requirement should be there. Getting? So land requirement in which the seed should be grown. That is the first objective. Next, it should be raised from seed whose source is approved. Getting? So source of the seed should be approved, and from that source you have to raise your seed, means you have to take that particular seed, and then field inspection should be done. The next third point, it should be provided with prescribed isolation. See, yesterday also I have discussed this. Isolation of that field should be there. Field should be isolated and or with the prescribed number of border rows in hybrid seed production. Suppose you want to take the uh, crop of hybrid seed, then you have to mention the number of the border rows, means number of border and how many rows are there, that should be mentioned before to do field inspection. So that should be done in case of hybrid seed production. Next point, that should be planted in a prescribed ratios of female seed and male parents. Getting? In case of hybrid seed how it comes, means you have to plant male seed, one row of male seeds and one row of female seed, and then the hybrid seed production is takes place. So here in a prescribed ratio you have to plant this female seed as well as male seeds. Next objective, it should be properly drugged to remove contaminating factors. So yesterday also explained about drugging. So this drugging is nothing but what? It is the removing of the plant of undesirable characteristics. So that is the rugging technique. So here again you have to remove those unwanted plant. Not only unwanted plant but the plant bearing the ears nothing but what? It is the head, or we can say that inflorescence which is converted into a seed. So here in case of grass it is particularly ear is the word is there. Getting? Ear. So here objectionable weed, again the weeds nothing but unwanted plant. So whatever the objectionable weeds would be there in case of that rows, you have to remove that one. Other crop plants are, if other crops also growing, you have to remove that crop plants. So means all these practice you have to done for field inspection. Then you have to make confirm that the particular variety is true to their characteristics. So particular variety for which you have to made the field inspection, it's going to show you the true characteristics of it. Means suppose you are taking one variety and means you are purchased one variety and you are going to do the inspection of another variety, that should not be happen. So particular variety is going to show you a true characteristics, so that is again a main point of field inspection. Next point, harvested properly to avoid mechanical admixture. Means you have to again harvested that particular crop, particular seed, and that should not be mixed with any other contamination, any other any admixture of within it. So it should be grown in a compliance with other special requirements for the crop consent, and other also very basic things which are needed, so that again you have to maintain for this field inspection. Next point, eligibility of crop varieties for certification inspection. So what are again eligibility criteria for doing this inspection? So again see, means suppose you want to take again admission. Suppose you want to take admission for graduation. What is the compulsory criteria? What is the compulsory eligibility is? You must pass 12th, right? So likewise, until and unless, means suppose you passed only 10th and then you want to do the graduation, that will not be happen, right? So likewise again, eligibility of that particular crop uh, should be uh, made, and then finally inspection should be done. So here see what are these eligibility criteria. So first is the certified seed crop must have been grown from foundation seed. Now see, in my very first lecture I have mentioned you, I have given you different type of seeds, right? What are the different type of seed I mentioned? So total five different seeds I have mentioned, that is nucleus seed, breeder seed, foundation, then certified and truthful seeds. All these five seeds I have explained you in detail. So please do remember the sequence or the types of that classes of seeds. Getting? So types or the classes of seeds that you must have to remember in a particular sequence, so that the progeny of the next seed you must have to remember this. So here see, certified seed crop must have been grown from foundation seed. Getting? So this is very important point, and that should be approved from a source recognized by certifying agency. So this certified seed must have been grown. Certified seed must come from whom? That must come from foundation seed, and that foundation seed should be approved from certifying agency. Next point, if they approved by a certification agency from certified seed, provided the genetic identity and purity will not be significantly altered. Means even if this certified seed is approved, their genetic identity and purity should not be altered. So that is again very important point. So here that should not be altered, right? Next, following seed classes should maintain their eligibility as follows. So now again, as per the classes of seed, you have to again look their eligibility criteria. Now which are those seed classes and what are their eligibility criteria? Now see the first type, breeder seed. Getting? So as I have mentioned you have to remember the type or the classes of seeds, so that you have to remember the particular seed come from which seed, right? So here see the breeder seed, the seed or the vegetative propagating material directly controlled by originating or sponsoring plant breeder of breeding program or institution. So here see, basically breeder seed comes from which seed? Breeder seed is the progeny of nucleus seed, right? Nucleus seed is the very first seed. Getting? So from the nucleus seed breeder seed is coming. Getting? So breeder said actually where it is going to grow or where it is going to come from. Breeding institution means research institution is there. In that research institute, this the means these breeder seeds they are going to raised, or breeder seeds are multiplied in research institutes. So here a particular plant breeder, that is researcher, they are doing this breeding technique of this breeder seeds. So here that should be happen in a particular institution. The next point, production of breeder seed is personally supervised by qualified plant breeder. Yes. So production of breeder is there, and that should be supervised by qualified plant breeder, because in a breeding technique you are going to uh, means cross two different plants, right? So that whole right from the breeding, right from till you are harvesting, all the stages of that particular plant should be supervised, should be observed by qualified plant breeder. Next, this seed provides the source for the initial and recurring increase of foundation seed. Yes, because see, from breeder seed, which further seed is going to raise? From breeder seed, your foundation seed is going to come, right? So that means it should, that breeder should be act as a source for whom? Source for foundation seed. So that you have to remember, because you must have again the quantity of breeder seed with you, so that foundation seed should be should be raised from these breeder seeds. Next type of seed is foundation seed. So here again see, in case of foundation seed, the progeny of breeder seed or foundation seed which can be clearly traced to breeder seed, means foundation seed again coming from which seed? Breeder seed. And if you don't know about this, means from which seed it comes, but again you have to uh, trace from where it comes. So ultimately breeder seed, the breeder seed from which the foundation seed is raised. So you are clearly going to trace this breeder seed by observing the characteristic of further progenies. So here second point, see, production supervised and approved by certification agency should be handled so as to maintain specific genetic identity and purity. So means all these conditions, their production of foundation seed, it should be supervised, and that should be again foundation seed approved by certification agency. And all these conditions, when it's going to certify this foundation seed, when the genetic identity and purity should be uh, maintained, at that time only this foundation seeds should be certified by that particular certification agency. Then third point of foundation seed is it should conform to certification standards specified for the crop. So again some standards, certification standards are there that should be fulfilled by this foundation seed, then only your inspection should be made in case of foundation seed. Next, come to the next type of seed, that is certified seed. Now what is the first criteria? What you have to inspect here in case of certified seed means where it comes from. So again I have mentioned you, certified seed, it comes from foundation seed, or the certified seed is the progeny of whom? Foundation seed ka ye progeny, right? Then next, its production should be handled to maintain specific genetic identity and purity. Again see this point, genetic purity and genetic identity, this is very important characteristics where these characteristics are repeating. Yes, because these are the standard seeds which are going to grown in a particular institutions only. So this genetic identity and purity you should maintain. Getting? So according to again standards, you have to maintain this genetic identity and purity. Then next point is, see here, inspection timing. What are the inspection timing and how many times you have to inspect the particular seed? Okay, so here see my first point, more than one inspection is done. Why this is so? Why this is more than one inspection is done? Why not only once? Because see, suppose you have visited the field only once, what the characteristics you are going to write there? Okay, whatever the things you are observing in a present, that characteristics only you have to note it down. You have to inspect only the characteristics which you are observing. Getting? So in a first sight you are not going to observe all the characteristics right from the seedling stage till the fruit setting stage, right? So here more than one inspection should be done so that you should have the knowledge about their seedling, about their pre-flowering stage, then flowering stage, then fruiting stage, then harvesting, then storage. So all these characters should be inspect at a different stages. It should not be inspect at only once. Okay, so that's why more than one inspection is done. Now, so inspection time depends on which of the characteristics. So these are a few characteristics I have mentioned here, so that you have to do your inspection at a particular stage. Now see, you have to first point you have to remember, that is crop duration. What is the crop duration? What is the life cycle of particular crop? So that duration you have to fix. Suppose particular crop is coming from January to March. So you have 3 months with you. So within that 3 months you have to visit that particular field many times. Many times means right from the sowing till the harvesting. So duration you must uh, you must have to know the duration of particular crop. The next, nature of pollination, means which pollinating agent is going to pollinate, whether it is wind, whether it is insect, or whether it is water. Means in nature of pollination you must have to remember of particular crop. Then susceptibility to contamination, means which contamination is going to occur within that field. Yes, you have a previous knowledge of that particular crop plant, which contamination, and then which uh, action you are going to take to control this contamination. So that thing again you have to mention or you have to remember. Then disease susceptible stage, which are those stage in which disease is going to occur. So this again very important point. Means suppose you know that in this particular stage this contamination is going to occur, means you are going to take a previous care in order to stop that occurring of disease. The next, nature of contaminating factors, or any single factor is there or many factors they are going to contaminate your crop. So that is the nature of contaminant factor you must have to know. Then other varieties, other variables, other factors they are responsible for your crop failure, that again you have to remember. Means all these conditions you have to remember while doing any inspection in any crop field. So all these characteristics, all these stages, all these important factors you have to consider while inspecting. Then for convenience, the stage of crop growth at which inspection are generally made in sexually propagated species are classified as follows. In my next slide, I'm going to show you at which particular stage of a crop you have to made your inspections. So this is the whatever the stages I have mentioned further, this is in case of sexually propagated species, right? So the further stages, see here which are those stages and what inspection you have to make, what you have to observe. Now first stage, see here, pre-flowering stage. Now in a pre-flowering stage means what? Before flower initiation, whatever the stage, that is pre-flowering stage, is very simple, right? So here what you have to look, what you have to inspect is this. You have to inspect about the seedling, what kind of seedling is there, how it is, whether it is strong, whether it is weak, how many leaves are there, how many birds present on it. So all these things you have to mention there. Then vegetative and flower bud initiation stage, again these things you have to remember, you have to learn, you have to make it note in your diaries, or one a particular field inspector should note it down all these characteristics. So all such growth phases prior to emergence of the panicle or inflorescence. Getting? So all these characteristics you have to remember in case of pre-flowering stage. In case of flowering stage, what you have to remember means when that panicle have opened. Getting means from the day of sowing till the opening, means you have to remember again the days when that flower spikelet. Now see here, spikelet is nothing but what? Spikelet. Here spike inflorescence is particularly for grasses. So all our these seed plants, they are very they are monocotyledons, they are all these are grasses particularly, right? So the spike means what? The flower which are attached without their pedicel on a uh, peduncle on a flowering axis. So that is nothing but that type of inflorescence is spike. So small these spikelets are present on that particular stem. So here particularly in case of grasses, this type of inflorescence present. So this knowledge also you must have while observing these flowering stages. And here again in flowering stage you have to note it down whether stigma is receptive or non-receptive, and again anther is shedding their pollen or not. So these three things means when it is open, whether stigma is receptive or not, and anther is shedding their pollen, when it is going to shed their pollen. So all these characteristics again noted down while doing inspection of these uh, cross pollinated crops. Then post flowering stage. Once the flower opened, then after flowering stage, post flowering stage is there. So in this stage what you have to remember is, here the receptivity of the stigma and pollen shedding of anthers have ended. In previous stage what we have learned, what we have to remember is, there is stigma is receiving the pollen, means both these things they are in uh, both the things means both the stigma and anther. Stigma is receptive and anther is shedding their pollen. So this stigma receptivity is stopped, and pollen also stopped shedding their anther. So it is ended, this stage is ended in post flowering stage, and fertilized ovule starts to develop into a seed. Yes, we know that once the pollen they are going to uh, attach to the stigma, the ovule of these uh, ovary, then these fertilized ovules they are converted into a seed. So this these things we we have this knowledge, previous knowledge. See the next point, post flowering stage. So these include both milk stage. Now see here, milk stage. So when the content of the fertilized ovule are in the form of white milky fluid, then it is called as milk stage. And dove, here it is dove. Dove stage is nothing but what? When the seed content are being transformed into a more solid pasty substances, then it is called as, then it is called as what? Dove stage. Getting? So dove is nothing but what? Here, here also we know that when we suppose we are having the wheat flour with us, when we are adding the water in it, in that wheat flour, we are going to form a dough, right? So here in case of these seeds, when the milky fluid is present, that milky fluid is begin to change to a duffy consistency. And why this duffy consistency is occurring? Because starch accumulation is going to happen within that endosperm. So milky fluid is somewhat going to convert into a dough stage. Getting? So here, so that stage is nothing but what? Dough stage. And dough stage recognized by having more solid pasty substance that yields to pressure. Now the next, pre-harvest stage and harvest stage. So here you have to remember that pre-harvest stage, here seeds become harder or reaches physiological maturity. Then seed is fully formed. Seed is now here fully formed, but it is high in moisture content. That is your pre-harvest stage. Seed which is having high moisture content. That is nothing but pre-harvest stage. In case of harvest stage, when you are going to harvest, when the seed become physiologically mature and sufficiently dry. At that time, we are going to harvest any particular crop, right? So this is very safest stage when any seed is sufficient dry, and it is also help in a threshing process, right? So this is very very important stage. You have to observe it. You have to inspect it. So in case of cross pollinated these species, or we can say sexually propagated species, I have mentioned pre-flowering stage, flowering stage, post-flowering stage, pre-harvest stage and harvest stage. Their specific points you have to remember, you have to inspect it. So that's how here the field inspection is done. And I have mentioned again different characteristics of and conditions of field inspection. So thank you for patience listening.